the spike protein, and we reached a resolution of 2.8 angstroms when we applied the symmetry, otherwise 3 angstroms. And the majority of the particles were actually in a close conformation, which was in previous structures usually a minority component. And when we started looking at the structure, we saw additional density, which you see here in the corner. So this additional density was definitely not protein. And it's really Christine Tölzer's achievement to actually look at this density, realize that in the unsymmetrized map, it's there in all the receptor binding domains, and it's surrounded by hydrophobic amino acids, mostly phenylalanine. And at this one end, where the carboxy group is, we do have a positively charged residue. So she decided that it is most likely a fatty acid, and then she went into the BDB and downloaded all the protein fatty acid structures and compared them to the density we had. And she decided early on that it's a C18 fatty acid, and it is most likely linoleic acid, because it is most similar to this fatty acid kinase and the odorant binding protein, which you see here. But of course, that's not a proof. And when she told us that this is linoleic acid, I decided to get in touch with our collaborators in Heidelberg at the Max Planck Institute. They are also part of our Max Planck Center for Minimal Biology in Bristol, Joachim Spatz and Oskar Stauffer, and they used LC-MS in order to show that the mass is indeed compatible with linoleic acid. And here you see the Coulomb surface, and you see that this pocket is really nicely hydrophobic, and the linoleic acid fits in like hand in glove. And in the meantime, we also analyzed second spike protein, which is a variant which has been discovered in Bristol. And we have done LC-MS with linoleic acid as the standard, and there we could show that indeed, yes, this is linoleic acid. Now, what happens when linoleic acid binds to this receptor binding domain? There was already a structure then from David Wiesler's lab, and so we compared our closed structure with the structure published by David Wiesler, and we found that there is a helix at the entrance of the pocket, which we named the gating helix, which moves about 6 angstrom in order to make space for linoleic to enter. And then there are also 2 tyrosines which have to flip away in order for a linoleic acid to bind. And there is the carboxyhydride group of linoleic acid, which is coordinated by an arginine and a glutamine. And these are provided by the neighboring receptor binding domain in the spike trimer. And this is accompanied by a compaction of the 3 RBD domains, and this is shown here in the right-hand corner, where you see how in this rather locked structure, the RBD domains move closer to each other. It's actually a 6 angstrom movement from the closed to this locked structure, which is also mirrored by the RBD domain. So altogether, this is much more compact. And we have now also really nice MD simulations from Martin Frank showing how linoleic acid actually can bind into that pocket and make this pocket opening. So as I said, the majority of our particles were linoleic acid bound, 70%, but 30% of the particles were in an open conformation, and the first spike structure we had was not sufficient resolution in order to decide whether there's any LA in there or not. This is interesting because the receptor binding domain is kind of tucked away with the receptor binding motif here in red. So this is not accessible for binding the human ACE2 receptor. So it's we consider this as a non-infectious particle, whereas this open position is required in order to dock onto human cells and bind to the ACE2 receptor. Here's the structure showing ACE2 interacting with the receptor binding motif. So in the meantime, we have solved also a SARS-CoV-1 structure, so virus that caused the first outbreak 20 years ago, where we had a bit more particles in the open conformation, and we could clearly show that there is indeed no linoleic acid bound to these ones. And the two RBD down domains are more like the closed conformation reported by David Wiesler. So in summary, what we see here is that when linoleic acid binds, it stabilizes a locked conformation, which is incompatible with infection, or vice versa. Linoleic acid has to first dissociate in order for the spike protein to get into an infectious conformation. And of course, we wanted to test this, and we started in vitro. And here's again a top view where you see how these receptor binding domains are tucked away and cannot interact with ACE2. And that's what we tested here with our spike sample, where about 70% is linoleic acid bound, and it still can interact, or the couple of particles which are open can interact with ACE2, which is immobilized on a biocore censorship. And we see here a nice binding curve. If we then take the spike protein and pass it over a lipidex column in order to remove the linoleic acid, we get a much better binding. And so indeed, there have been particles which could not bind to the surface. And linoleic acid binding interferes with the receptor interaction. We also tested this with live SARS-CoV-2 virus. We are in Bristol in the nice situation that we actually had somebody working already on SARS-CoV-1, Andrew Davidson, and in the last 2 years, he became the most sought after person in Bristol, I think. So he, together with and Maya Kavanagh-Williamson, they actually tested whether if we add linoleic acid to infected cells, whether this changes anything in the infection pattern, and indeed he does. So he had GFP expressions in SARS-CoV-2 virus, and this has been just treated with remdesivir, which at that time was the only drug which was known against COVID. And you can see that you need quite a lot of remdesivir to actually stop viral replication. If we add on top of this 50 micromolar linoleic acid, then you have many fewer green virus infected cells here. So altogether, the viral replication is inhibited. And this is also shown here with the quantitative RT-PCR, where the viral genome is determined. And you can see that already at 20 nanomolar remdesivir, 50 micromolar linoleic acid, we see a clear decrease in viral replication and RNA. So in the meantime, Oskar Stauffer also has tested this in another way outside of the CAT3 lab. He generated these small unilamellar vesicles, which have the same size as SARS-CoV-2 and have the same lipid composition from the, basically he took the composition from the ER to make these vesicles. And part of the lipids are labeled with a fluorescent dye. Other parts of the lipids have a his tag, nickel anti-A, and so we can use our his tag spike protein to actually immobilize it. You see here slices from a tomogram, and indeed there are spike proteins immobilized on these vesicles, and we can nicely use these for fluorescence imaging in order to see whether these particles, which are not replicating, obviously, because they are entirely synthetic, so they bind on the surface of these MSC7 epithelial cells, and we can quantify this. And we have normalized it here against native spike. So that's our spike from insect cells, which contains about 70% of linoleic acid bound spike proteins. If we remove that and have the ApoS, which is not linoleic acid bind, you see a better retention of these SUVs or mini variants on the surfaces. And the same happened when we tested different free fatty acids, including palmitoic acid, which is also C18, that has no effect, but interestingly, all the polyunsaturated fatty acids, linoleic acid and arachidonic acid, kind of interfere with the binding of the particles to the surface. So again, clearly that interferes with docking of the particles on the surface. But then Andrew said something interesting when he did these original experiments with remdesivir. He said that even the green cells which were treated with linoleic acid were actually looked more healthy. And that is, for me, that was like, what do you mean more healthy? How can the cells look more healthy? And he couldn't really explain it. So in the end, we decided that we should look into the cells. And together with Paul Verkade and Lorna Hodgson in Bristol, we performed CLEM experiments to look into the cells. And here is the control experiment where we have CARCO2 epithelial cells which express ACE2 and which we infected with this GFP expressing SARS-CoV-2 virus. And then because we wanted to look only at infected cells, which apparently looked healthier, we decided to treat the cells only 1 hour after the infection with 50 micromolar linoleic acid. So the control experiment is shown here, and you have seen much nicer images of infected cells from Louisa on day 1. But again, we have all these vesicles, these viral vesicles induced here in the cytosol. We focus here on the vesicles which are closer to the periphery of the cells and which are full of already assembled variants. And if you go in here in the close-up view, you see these variants. And this, when you go into the treated linoleic acid treated cells, you have also these vesicles, but there are fewer of them, and also we find many fewer variants in these cells. What was striking me first, most at the beginning were these big dark blobs, and some of them were really huge, and so I was a bit frightened. But then when we did the same experiments with non-infected cells, they also showed these dark blobs. And then actually I found in old histology books images of adipocytes. So these are lipid droplets. So we treat ourselves with linoleic acid, and clearly they take up the linoleic acid, and here we find lipid droplets in the cytosol. So nice internal control that linoleic acid really is in there in the cytosol. And what we observed altogether with the microscopy analysis, that there were fewer green cells, even though we can't quantify that yet. We are in the process of repeating the experiments with larger volumes. But what we can quantify is that there are less variants per infected cells when we treat with linoleic acid. And so on average, we have in these cells 25 particles per micrograph, which is approximately a cell. And then we have 9 particles for the linoleic acid treated cells. And not only were there fewer variants, but they were also larger and deformed. So here's the size difference between them. So something is wrong, or something is wrong when, or goes wrong for the virus when linoleic acid is there. And this is where a action of linoleic acid starts, which I'm not even sure whether it has anything to do with the spike protein. But what is known is that when the virus infects cells, it basically hyperactivates cytosolic phospholipase A2. And so we talked to Gérard Lambeau and Christine Berger at the University of Nice, which works since many years with CPLA2, and asked them whether they would actually test the effect of linoleic acid on CPLA2. Since it's known that CPLA2 is hyperactivated, it is also a drug target, and there are already several drugs known against CPLA2, so they are in this experiment shown here. It's also activated by divalent ions, so with EDTA you can inhibit the enzyme nicely. And if we add linoleic acid to CPLA2, basically we see less fatty acids released. And that is basically a feedback loop in the cell, that when CPLA2 is activated and enough fatty acids are in the cytosol, that basically then the enzyme is shut down again. And somehow the infected cells circumvent this feedback loop. And I don't think that there's really enough spike protein in the cell to circumvent this, given the inhibition constant, which is around 100 micromolar, but clearly it needs these fatty acids in order to generate all these new membranes in the cell. And so I think where these fatty acids end up are in the viral membranes. And so they can't really inhibits CPLA2, and CPLA2 really continues to produce new fatty acids for the virus. Now, switching gears, another important question is obviously how conserved is this pocket? And here is a list of all the beta coronaviruses that infect humans. And if you look at these gray residues, these are the ones which line the hydrophobic acid pocket. And they are all conserved among the different beta coronaviruses. And for several of the viruses, also these positively charged residues, which coordinate the carboxyl group, are present. So based on this, we hypothesized that the pocket is conserved, and we also solved the structure of SARS-CoV-1 and showed that it is conserved since at least 20 years. And the good news is that it's also conserved in Omicron and all the other variants of concern. So actually, Kapil in our group purified all these RBDs, and I'm not going to show you all these biocore curves, but just the three most interesting facts we found. So here's Omicron. It binds to the RBD of Omicron binds linoleic acid with 40 nanomolar affinity, and that's just the RBD, not the entire spike protein, where you potentially have additional cooperative effects, and where you also have the coordination of the head group. So the real affinity is higher. This is just the lower end of the affinity. And here's the structure of Omicron with all the mutations highlighted in red. And they are interestingly all away from the free fatty acid binding pocket. And that's true for all the other variants of concern as well. Then there are two really interesting cases. So there is this beta coronavirus HKU1, which causes a common cold. And in the structure here, we have highlighted the residues which are identical to SARS-CoV-2, but this one has a glutamate in front of the pocket. And indeed, in biocore, we don't see any binding. But if we remove the glutamate and replace it with an alanine, we restore linoleic acid binding. So we are one mutation away from a linoleic acid binding HKU1, which has already a decent affinity, 180 nanomolar. When we published our first paper about the pocket, we found that OC43 has already this locked structure according to the PDB file. And we said, well, it probably will not bind linoleic acid because it's somehow locked. And then David Wiesler, actually, who published this structure, contacted us and said, well, you know, we used beta-octylglucoside to prepare the grids, and then we saw some density there, and we thought it's the detergent, and we really didn't know what to do with this. But actually, there's density in the pocket. And here, indeed, here it is. This is his EM map. It's not linoleic acid. That's an overlay with our structure. And just last week, I got an email from a friend who actually forwarded me this paper from bioarchives, which has analyzed OC43 with LCMS, and they found that it is sapienic acid. So again, a fatty acid with an unsaturated bond, but it is C16. And it really nicely fits this density. And that's an interesting choice for OC43 versus SARS-2, because actually sapienic acid, I had to look this up in Wikipedia. It's one of these fatty acids which only humans make, and it's very abundant. So it seems that in SARS and MERS, the highly pathogenic coronaviruses, they specifically bind linoleic acid, whereas HKU1 can live without any free fatty acid binding, and OC43 takes one, which is very, very common. Whereas this binding pocket, linoleic acid, is highly conserved in all the different variants of concern. And so our hypothesis now is that there is a correlation with the high pathogenicity and linoleic acid, but that's of course not easy to test because you can't do that in cell culture. So we will need animal models for that. So what's so special about linoleic acid? First of all, it's a vitamin. It's the one fatty acid, one of the two which we as humans can't make ourselves, vitamin F. Normally you don't bother about it, but actually in severely sick COVID-19 patients, the linoleic acid levels in the blood serum are decreased. And I already kind of said this, that linoleic acid is actually in the membranes, and it is as a polyunsaturated fatty acid. It is very important to maintain the membrane fluidity, and particularly important in lung cells, where it is helping to maintain the surface tension. And it has been shown before COVID that any alteration in the linoleic acid pathway lipid composition is associated with acute respiratory distress syndrome and severe pneumonia. And both obviously are key elements in COVID-19 pathology. And this is really thanks to our colleague Dan Fitzgerald, that we know all of this, because he then pointed out that the linoleic acid in the body is metabolized to arachidonic acid and then eicosanoids, including prostaglandin. And these molecules regulate inflammation and the immune response. And again, this is severely disturbed in humans, in COVID-19, as we see that even the fact that the serum levels go down. And Dan worked for more than 10 years on the free fatty acid binding receptor, which is a key regulator of insulin release central to diabetes. So interestingly, a free fatty acid regulates insulin glucose metabolism. And as you know, diabetes is a risk factor for COVID-19. So there are many reasons why LA is a really bad choice. And I think what we found here is a structural link between linoleic acid, the pathology, and the virus itself. And of course, it suggests that one could exploit this linoleic acid binding pocket, and also the justice signaling axis in order to intervene and have an intervention point against SARS-CoV-2. So I've shown you that there is an LA binding pocket in SARS-CoV-2 spike protein. LA stabilizes a non-infectious form of spike with the RBDs locked down. And then I didn't have time to talk about this via allosteric effects, and it has been analyzed a lot by MD simulations in Adrian Mulholland's lab in Bristol. LA binding also translates all through the molecule and also affects the furin cleavage site and the fusion peptide proximal region. That's where the proteases cleave in order to switch from the pre-fusion to the post-fusion state of spike protein. And I think we showed that LA reduces the viral docking onto cells and thus the infection into the cells, and inside the cells the replication. So of course that requires more research. And we think this can be developed into a novel potent treatment of COVID early on. And so what we did last year was actually start developing a nasal spray such that you could early on, when you are exposed to SARS-CoV-2, or even as a prophylactic, use linoleic acid and free fatty acid combination in order to interfere with viral replication. And now this is work which I'm only following, but it's extremely interesting. So basically then Fitzgerald developed a product, and I never thought that this is so much work, that actually here's the drug, so here's the product. No, this is not how it is. Actually, you need to think about shelf life and how to stabilize it, how to prevent oxidation. So he actually worked with GMP suppliers. He worked on the formulation, which active ingredients to include, and testing different devices. So not every nasal spray gives you the same dose. Sometimes it's like this: if you push harder, you get a higher dose. That's maybe not a good idea. You always want to have the same dose. And this is now has been finished, and we are entering a combined phase 1/2A trial, where initially 16 healthy patients, not patients, healthy beings, human beings, will test this nasal spray. And that is work which is done in the Bristol Medical School by Adam Finn and Catherine Himes. And they developed the clinical phase trials. And with this, I hope I mentioned everybody who was involved in that project on the way. And these are all the people in IMRIS and my group and all the significant others. And thank you very much for your attention.
And it's really puzzling because you show that the linoleic acid is locking the spike, yet that drug binding pocket is conserved. And there is linoleic acid in the cell. So how can that puzzling fact be explained? Because why if it's like 20 years, why there is no pressure for the virus to mutate that site?